All right, AP Biology, this is our Chapter 7 on photosynthesis. A lot of this you may have uh, studied before in a previous biology course, especially advanced biology. Some of you may be introduced to some new things in here. There are electron carrier molecules and cytochrome complexes that we'll learn about as well. But just to understand, as a basic component of what photosynthesis is, why is it an essential process for living things on Earth, and what are the two main reactions that are carried out in what we would call photoautotrophic organisms? Remember, photoautotrophs, or an autotroph in general, is an organism that makes its own food. So, and the fact that we are looking at photosynthesis, we have what we would see photoautotrophs, which are green plants, cyanobacteria, algae, those all photosynthesize. So we have photosynthesis occur on land. We also have it occur in our lakes and ponds and within the ocean itself, as long as light is available for the reaction to occur. And in the ocean or in a lake, we would call that the photic zone. So basic outline, in 7.1, it's the photosynthetic organisms. 7.2, the process of photosynthesis. 7.3 is how plants are going to convert solar energy. And then we're going to look at, in 7.4, plants fixing carbon dioxide. And then we'll look at the various types of alternative pathways in section 7.5. So basically, photosynthesis as the key to solving our fuel crisis. One of the things plant scientists or botanists are tweaking the basic chemistry of photosynthesis to create commercially important oils and fuels. There you have an example is camelina. It's a drought-resistant oilseed crop. And by using genetic engineering, scientists are improving the efficiency of photosynthesis and improving the CO2 absorption rate to increase the raw material for oil production. Another example is terpene, a high-energy organic molecule from pine trees that is used to make turpentine. So all life depends on solar energy. I would say it's probably about 99.7%. I think I remember seeing a figure. 99.7% of all life on Earth does depend on photosynthesis for survival. Whether it be you are taking in oxygen gas as the byproduct, or it begins the basis of most food chains on this planet. It's those photoautotrophs. The exception to this would be the chemoautotrophs, and those organisms live in the ocean deep. They are way beyond the photic zone of the ocean. So what they do is they use chemosynthesis, chemical synthesis, using that hydrogen sulfide gas from the hydrothermal vents to convert that to organic molecules. So we know that photosynthetic organisms are algae, plants, and cyanobacteria. Photosynthesis is a process that captures solar energy and transforms solar energy into chemical energy. And the energy ends up being stored in a carbohydrate. If you could think about the gummy bear, when it was put into that potassium chlorate, all that energy that was released from that gummy bear, and all the sugars that are in that gummy bear. So you can see that you do get quite a bit of energy out of glucose. Photosynthesizers produce food energies that is used to feed heterotrophs, which are consumers. So photosynthetic organisms, photosynthesis takes place in the green portion of plants. This would be the part of the plant where the chloroplasts are found. And if you recall, the chloroplasts are those specialized organelles that are going to be carrying out photosynthesis. Usually you have an abundance of chloroplasts in a tissue called the mesophyll tissue. We'll look at this a little bit later. but the mesophyll tissue is where chloroplasts are found, and there's typically a greater abundance of chloroplast in the palisade mesophyll. Basically, the raw materials for photosynthesis are CO2 and water. Roots absorb water that moves up the vascular tissue. Remember, water would travel via a plant, the xylem tissue, Xylem, that's pronounced xylem, and it's going to be traveled up to the leaves. Carbon dioxide is going to enter into the leaves through small openings called the stomata. Remember that those stomata are the small openings, and their opening and closing of those stomata are controlled by guard cells, and that is going to be the basic process of simple diffusion. And then we have the reaction within the chloroplast. The one type of reaction would be the thylakoid membranes of chloroplast. The thylakoid membranes are where we find the chlorophyll, as well as other pigments, but chlorophyll is the main photosynthetic pigment, and there are 4 types of chlorophyll. And then we have the carotenoids, the xanthophylls, the carotenes, betalains, all those. Electrons are energized in the process. So remember that when light energy is absorbed, electrons are going to go from their ground state to an excited state where they are unstable, and eventually they will fall back down to release energy and heat. Or, in the case of photosynthesis, they will travel through the thylakoid membrane, and that would be the process of the ETS system, the electron transport system. The second reaction is take place in the stroma. The stroma combines carbon dioxide with water to form or sugar. So this is where the synthesis portion of photosynthesis comes into play. So the thylakoid membranes, where chlorophyll is going to harness light energy, that's where photo occurs, and synthesis occurs in the stroma. And the stroma is an aqueous compartment. It's the bulk volume of the chloroplast, and it also has the enzymes which are capable of carrying out, speeding up these reactions to make that sugar. So here you could see examples of those organisms and the relationship between a photoautotroph and a heterotroph. Here's the typical leaf structure and chloroplast structure. The upper layer here is the cuticle. That is a waxy layer of the leaf. That waxy layer of the leaf is going to help prevent water loss. Then we have the upper epidermis. The upper epidermis and both the lower epidermis lack chloroplast. And then at right angle to the upper epidermis are these cells called the palisade, Palisade. These are the palisade mesophyll cells. And then below that you have what you would refer to as the spongy mesophyll. The palisade mesophyll typically have a greater abundance of chloroplast than the spongy mesophyll because light energy is going to be striking that upper surface of the leaf. But both cell types do have chloroplast. Why do they call it the spongy mesophyll? That is because there are air spaces in here, and the air spaces are where we're going to have our gases to be able to come in and diffuse in and out of this leaf here. Also in that part of the leaf, we do have this vein, or what we would call the vascular bundle, and in there we have both of our conducting tissues in the plants, the xylem tissue, which is the water conducting tissue of the plant, Xylem, and then we have the phloem tissue, Phloem. The phloem tissue is the food conducting tissue of the plant. And then right here in the lower surface of the leaf, you can hear it, we have the stomata, or singular stoma, and those 2 little specialized cells, which are the only cells in the lower epidermis to contain chloroplast, are what we would call guard cells. And basically we look at the flow of potassium ions in and out of the special compartments of those guard cells, and how osmosis will help those cells open and close. So if we go then in further, here is the chloroplast. Remember, chloroplast once thought to be their own separate living thing. So when they came into the eukaryotic cell, they got a second membrane. So we have both an outer membrane and an inner membrane there, according to the endosymbiotic theory. We do have the stroma, which is that large compartment of water and enzymes. the bulk volume of the chloroplast, and then we have these thylakoid stacks. And these are membranes that are going to contain the chlorophyll and other pigments to harness light energy. Basically, a pigment is a light harnessing substance. You see there that a stack of thylakoids, which looks like a stack of coins, is what we refer to as granum singular, or you could say grana, Grana. All the grana, which you can see here, are the stacks of those thylakoids. And you can see that those are connected. So there are channels between one stack and another stack. So the process of photosynthesis, there are two, the light reactions and the Calvin cycle. The light reactions take place only in the presence of light energy. The energy is going to be used to energize electrons, and electrons are going to move down an electron chain. And as it moves down the electron chain, hydrogen ions are going to be pumped into the thylakoid compartment. And then electron transport chain is used to make ATP out of ADP, that's chemiosmosis, and you get an electron carrier out of there called NADPH. NADPH is formed from NADP. In the Calvin cycle, which takes place in the stroma, we're going to reduce carbon dioxide is reduced to form the carbohydrate, and the reactions use ATP and that NADPH made from the light reactions to help drive that process. So ATP is going to supply the energy, and NADPH is going to bring that electron in to the cycle, because that is an electron carrier molecule. So NADPH is going to be oxidized and CO2 will be reduced. The reactions themselves were named after Melvin Calvin. He used carbon isotopes to trace carbon throughout the process of photosynthesis. And basically, photosynthesis involves a series of oxidation reduction reactions. Remember that oxidation is the loss and reduction is the gain of electrons. So in the very beginning of this process, water will be oxidized in the light reaction, and CO2 is going to be reduced in the Calvin cycle, or the carbon fixation reactions. So just a quick overview. There is the thylakoid membranes. Solar energy will be harvested in, striking that chlorophyll pile to excite electrons. We also have the splitting of water, so water will be oxidized, and from there we'll get in NAD plus and NADP plus, and out of there we'll get oxygen gas as a byproduct of the splitting of water. We will get our electron carrier molecule NADPH, and we'll get energy in the form of ATP. And both these will be used in the Calvin cycle, which takes place in the stroma. Carbon dioxide is going to be reduced when NADPH comes in with the electrons. And ultimately what will happen is these two molecules are going to supply the energy that is needed to take carbon dioxide and form a molecule of, well, what we would say is sugar. But we typically get out glyceraldehyde 3-phosphate, G3P, from this process. And G3P can be used to synthesize other macromolecules as well. So basically, the plants convert solar energy, that would be pigments in the photosystems. Wavelengths that are not absorbed by pigments are reflected or transmitted. This is especially true for green light. In the visible light spectrum, green light is reflected or transmitted, and that's why we see the color green. Within that absorption spectrum, the pigments found in chlorophyll absorb various portions of that visible light spectrum, and an absorption spectrum basically is that graph that's going to show that relative absorption of the various colors of the visible light spectrum, which would be the colors of the rainbow, that Roy G. Biv: red, orange, yellow, green, blue, indigo, and violet. You do have accessory pigments. Accessory pigments do absorb other wavelengths of light, and basically that's so it doesn't, those other wavelengths of light don't cause damage to chlorophyll. So some of those would be the carotenoids, which are your nice oranges, yellows. Um, you have the violets, typically betalains. Carotenes are the orange light. The xanthophyll pigments are your yellow light, or yellow pigments, I should say. So if you look here, here is the electromagnetic spectrum itself. And you know we have gamma rays, X-rays, UV, and then we have in here the visible light spectrum. And it goes infrared, microwave, and radio waves. And as you go from gamma rays to radio waves, you are going to increase the wavelength, or increase lambda, is the Greek letter used to represent wavelength. And if you go the opposite direction, from radio waves to gamma rays, you are going to increase the energy. So if we think of a radio station transmitting its signal from city to city, they would have very long wavelengths. But over here, we know that X-rays, they are high in energy. That's why you can only get a few X-rays within a certain amount of time period, as fear of the energy and the exposure to radiation could lead to cancer. But the area that we are most focused on with the process of photosynthesis is right here in the visible light spectrum. And if we look at that absorption spectrum for photosynthetic pigments, we do see where those colors do have relative absorption. So we do see the curve for chlorophyll A. That would be the nice main photosynthetic pigment. And then we have chlorophyll B, which is right here. And then we have the carotenoids, which are right there. So we can see the colors that these pigments do absorb. And basically the relative absorption is very low right here in the colors of green. And that's because chlorophyll is going to reflect or transmit that green light. So plants do convert solar energy. The light reaction consists of two alternate electron pathways. We have the cyclic and the non-cyclic pathway. The non-cyclic pathway is the main reaction, or what we'd say the light-dependent reaction. The cyclic pathway is where we're going to see electrons be excited and then fall back down to their ground state, and that's where we get the emittance of heat and light energy. When you extract chlorophyll out of, for say, spinach leaves, and you beam light on them, you could see the cyclic pathway occurring there. What will happen is the chlorophyll will fluoresce, and it will change like a dark reddish, bloodish color. The light reactions capture light energy with photosystems. There are two, photosystem 1 and 2. Photosystem in general is just a pigment complex. So now this is something new. Basically, a photosystem is that pigment complex that helps collect solar energy like an antenna. The photosystems are located in those thylakoid membranes, and both the cyclic and non-cyclic pathways produce ATP, but it's the non-cyclic pathway that is going to produce NADPH. So basically what happens in the non-cyclic pathway, this is the light-dependent reaction of photosynthesis. It takes place in the thylakoid membranes. It uses photosystems 1 and 2. Photosystem 2 captures light energy. The non-cyclic pathway begins with that photosystem, and basically that was because that photosystem was discovered after photosystem 1, but we realized that it actually, where all the excitement occurs for photosynthesis, it is at photosystem 2. So what's going to happen is it's going to cause an electron to be ejected from the reaction center, which is chlorophyll A, in there, and the electrons are going to travel down the electron transport chain to photosystem 1. The electrons there are going to be replaced with an electron from water. Remember, water is oxidized. And you also generate oxygen gas and hydrogen ions. This causes hydrogen ions to accumulate in the thylakoid chambers on the inside, and that is going to be used to establish a hydrogen ion gradient that is used to produce ATP. In photosystem 1, basically what we'll have is the photosystem will capture light energy and eject an electron, and the electron is going to be transferred permanently to a molecule of NADP+. Remember, wherever that electron goes, so does what I would say is the wife, which is that hydrogen ion. So if NADP+ comes in, it picks up the electron, it picks up a hydrogen, and we form NADPH. NADPH is a high energy electron carrier molecule. And we could see all that happening here. We'll look at a diagram in class as well. So here we have water being split. We generate 2 hydrogen ions, half an oxygen gas molecule. Photosystem 2. Electrons get excited when solar energy is absorbed. And then it's going to be captured in that electron acceptor. So you have the primary electron acceptor, plastoquinone. We travel down the electron transport chain. So as that electron moves down the electron transport chain to photosystem 1, it's going to release its energy because it's going back to a ground state. In doing so, we will move hydrogen ions across the gradient there. Remember that the electron transport chain itself is a series of membranes that is going to use high energy electrons to transport those high energy electrons, releasing their energy to move hydrogen ions across the membrane from areas of low to areas of high concentration. In photosystem 1, the electron is back at its ground state. Solar energy is striking that pigment pile complex, and the electron is going to get accepted again by an electron acceptor when it becomes excited. But instead of falling back down to its ground state, what we'll do is release that electron into NADP+, where it's going to pick up the electrons and the hydrogen ion to form NADPH. And the ATP and the NADPH are now going to be used to drive the Calvin cycle, or what we'd say is the light independent reaction of photosynthesis. So PS2 consists of a pigment complex and electron acceptors. They receive the electrons from the splitting of water. Oxygen gas is released. We do have the electron transport chain. The electron transport chain itself consists of cytochrome complexes and plastoquinone. Those are going to carry the electrons between PS2 and PS1, and it also pumps hydrogen ions from the stroma into the thylakoid space. So that is something that you would want to know now. In the ETC system or ETS system, you do have things called cytochrome complex. And look at that word, cytochrome. Cyto means cell, chrome means pigment. So yeah, these cytochrome complexes and this acceptor molecule called plastoquinone. PS1 has a pigment complex and electron acceptors adjacent to an enzyme that reduces NAD+ to form NADPH. ATP synthase is the molecule that, coupled to ETS, you have chemiosmosis, and ATP synthase is going to have a channel for hydrogen ions to flow through, and that's going to be used to phosphorylate ADP into our high energy molecule of the cell, ATP. And you can see that process taking place right in here. So here are all those cytochrome complexes. There is plastoquinone. Plastoquinone is abbreviated PQ. And you can see that as those electrons are traveling down, we are moving those hydrogen ions into the thylakoid compartment, which is very more on the acidic side due to all these hydrogen ions in here. And basically those hydrogen ions will then pass through ATP synthase. So here we have the illustration of the ETS system, and here we have chemiosmosis, which you can see spelled right there. So these hydrogen ions are going to pass through this enzyme, ATP synthase, and convert ADP to ATP. So plants as solar energy converters, the thylakoid space acts as a reservoir for hydrogen ions. Each time water is oxidized, 2 hydrogen remain in that thylakoid space. The transfer of electrons in the electron transport chain yields energy via chemiosmosis and the ETS system. So the ETS system is going to pump those hydrogen ions across the thylakoid membrane, and then the proteins move from the protons, or what we'd say is the hydrogen ion, that's also referred to as a proton. The protons are going to move from the stroma into the thylakoid space as those electrons are passing through. The flow of hydrogen ions back across the membrane energizes ATP synthase to catalyze the reaction to form ATP. So tropical rainforest destruction, a little bit of ecology and environmental science thrown in here. Tropical rainforest destruction and climate change. Tropical rainforests can exist where temperatures are above 26 degrees Celsius and rainfall is rather heavy, ranging from 100 to 200 centimeters and regular. So what we see here is most tropical rainforest plants are woody plants with many vines and epiphytes. Epiphytes are air plants. They typically grow on other plants. There is little or no undergrowth. So when you talk about the undergrowth, that would be the plants that are on the forest floor. Tropical rainforests contribute greatly to the carbon dioxide uptake, which basically slows global warming. Development has reduced the rainforests from 15% to 5% of Earth's surface. And basically deforestation itself accounts for 20 to 30% of the atmospheric CO2, but also removes that CO2 sink. So the burning of fossil fuels adds CO2 to the air, and increasing temperatures also reduce the productivity there. And if we look tropical rainforest destruction and climate change from the 1900s up to the 2100, here's the year 2000. These are projected across the x-axis. We do have the date in years. Across up and down on the y-axis, we have the global surface warming in degrees Celsius. So we do see basically what our minimum would be at 1 degree Celsius. Still very brings a lot of concern. Just raising that global temperature by 1 degree Celsius is a lot of concern, but there's also projected that if we keep going, we could actually raise it up to a little under 4.5 degrees Celsius, and that would have a very drastic effect on our planet. So you know some of the things that are discussed with climate change, and we could discuss that a little bit further in class. In 7.4, plants are going to fix carbon dioxide. So here's where we're going to see the Calvin cycle. It's a cyclical series of reactions that utilizes atmospheric CO2 to produce carbohydrates. It's also known as the C3 pathway because CO2 is going to be fixed into a 3-carbon compound. It involves 3 stages: carbon fixation, carbon dioxide reduction, and the regeneration of RUBP. So basically, carbon dioxide is attached to a 5-carbon RUBP, ribulose bisphosphate, by the enzyme RUBP carboxylase. RUBP carboxylase, also sometimes known as Rubisco. It results in a 6-carbon molecule. This splits into two 3-carbon molecules called 3PG. And basically the reaction is accelerated by RUBP carboxylase or Rubisco. CO2 is now fixed because it is part of that carbohydrate. So once carbon dioxide gets reduced, we have that CO2 that is going to be fixed in those molecules. So if we look here at the cycle, we see ribulose bisphosphate or 1,5-bisphosphate. So we have 3 molecules of RuBP coming in. Those each have 5 carbons. And we have 3 CO2 molecules coming in. And we have that intermediate there. And basically what's going to occur there is, here's where, right in here, is where we have that enzyme doing carbon fixation. So basically what we end up with is 6 molecules of 3PG. Over here you can see PG is phosphoglycerate. Those are each 3 carbons apiece. And then now we have our oxidation reduction step and energy supplement from ATP and NADPH. So basically, phosphoglycerate, 6 molecules of phosphoglycerate with 3 carbons. Here we get each of those molecules is going to get phosphorylated and supplied with energy to do the conversion from here to an intermediate molecule. And then when NADPH, 6 molecules, 1 for each molecule here, still at 3 carbons, each NADPH is going to come in and it's going to be oxidized and reduce this compound to form 6G3P. This is glyceraldehyde 3-phosphate. That is the molecule that is going to be used to help make the sugar. So ultimately what happens is now you lose 1, and that 1 can go on to make other organic molecules or go on to make a carbohydrate such as glucose. The other 5 are going to continue back in the cycle. Here's we're going to regenerate RuBP again, and in order to do so, what we need is energy. So we get 3 ATP molecules that are going to come in and convert those 5 G3Ps, each with 3 carbons, back into 3 molecules of ribulose bisphosphate, each having 5 carbons there. So here we see carbon fixation using RuBP carboxylase or Rubisco. Here we have CO2 reduction. So we're going to reduce it by adding electrons in, especially from NADPH here. And now we have the regeneration of the starting reactant, which is RuBP, and we need to use energy there. Remember that this cycle needs to occur 2 times. Each time you're bringing 3 CO2s, so there's where you get your 6 carbons from. So this step does occur twice. So the first time we get out 1 G3P, which is 3 carbons. The second time out we get another 1 G3P 3 carbon compound, and together we can make those into a 6 carbon glucose molecule or convert it into other organic molecules. So plants fix CO2, 3PG is reduced to BPG. BPG is then reduced to G3P. Electrons and energy are required for this stage. This utilizes NADPH and some ATP produced in the light reaction, and glyceraldehyde 3-phosphate is going then to be reduced and chemically able to store more energy and form larger organic molecules such as glucose itself. The reduction of carbon dioxide ultimately then is summarized. We have the 3PG to G3P here. So here's where that ATP comes in. Here's that NADPH comes in. Both of those are supplied to the Calvin cycle from the light-dependent reaction where they are made. In the regeneration of RuBP, ribulose bisphosphate, it is used to fix CO2 and it must be replaced. So every 3 turns of the Calvin cycle, you get 5 G3P molecules that are used to make 3 RuBPs. So 5 times 3 equals 3 times 5. And ultimately there we have that regeneration there. As 5 molecules of the G3P become 3 molecules of RuBP, 3 molecules of ATP become 3 molecules of ADP plus P. And that's basically what they were summarizing here. So here you're going from 3, and here you're having 5. So it does balance out here with the number of carbons. So we have 5, 3 carbon molecules of G3P. Over here we have 3, 5 carbon molecules of RuBP. So plants fix CO2. We have glyceraldehyde 3 phosphate, can be converted to many other organic molecules. Basically, the hydrocarbon skeleton of G3P can form fatty acids and glycerol to make plant oils. Be used to make glucose phosphate, simple sugar, fructose, starch, cellulose, and amino acids. So technically, we're not just making sugar here. We can make other things as well. But we do look at it as basically the food for autotrophs and heterotrophs on our planet. Lastly, alternative pathways. The majority of plants do carry out the C3 photosynthesis or the C3 pathway, the Calvin cycle. These are going to use ruby P carboxylase to fix CO2 to ribulose bisphosphate, and this will take place in the mesophyll cells. Remember, meso means in between, so the mesophyll tissue or the mesophyll cells is tissue that is found between the upper and lower epidermis of the leaf, which was discussed in the beginning of the chapter. In hot, dry climates, stomata must close to avoid wilting. CO2 decreases and oxygen will increase, and oxygen starts to combine with ruby P, leading to the production of carbon dioxide. This process itself is called photorespiration. C4 plants solve the problem of photorespiration basically by fixing carbon dioxide to PEP, which is a C3 molecule, PEP carboxylase. The result is a compound that is called oxaloacetate, which is a 4-carbon molecule. Oxaloacetate basically will then later be able to release that CO2 molecule. So in hot, dry climates, the C4 compound, oxaloacetate, is going to be used to avoid photorespiration, which is what would happen up in here. Basically, the net productivity is about 2 to 3 times greater than that of the C3 plants. In cool, moist environments, C4 plants can't compete with C3 plants. So basically what we have is here is the C3 plant pathway. This is what we normally see here. Look at the arrangement of the mesophyll cells. Look at the arrangement of the mesophyll cells here. So we get all these cells that are kind of wrapped around the vascular bundle here, or the vein of the plant. So these would be at right angles in the C4 plant. So what we see is fixation in the C3 pathway. Basically, CO2 would come in to the mesophyll cell, be converted into the PG molecule, and then that would be converted to G3P, and then ruby P there. So we have ruby P fixed with CO2 to form phosphoglycerate, which is then formed to glyceraldehyde 3 phosphate. But here, what we have in the C4 pathway, which is common of corn, instead of having it all take place in the mesophyll cell, we have carbon fixation take place right here in the bundle sheath where it will be stored. So this process will take place in the bundle sheath. Again, that would be this area. The bundle sheath or the vein of the plant will be out in this area. So CO2 comes in, it's converted to that 4-carbon compound, oxaloacetate. So that's where we have the carbon fixation occurring in the mesophyll cells. And then that C4 organic acid will then be releasing CO2 into the bundle sheath where the Calvin cycle will occur. So this is a spatial separation occurring in 2 different types of cells. We do have CAM photosynthesis. This will be a temporal separation, so day and night. Basically, CAM photosynthesis stands for Crassiolnaceae. or crassulacean acid metabolism. Crassulacean acid metabolism is partition carbon fixation by time, so it's temporal here. So during the night, CAM plants fix CO2, forms the C4 molecules, which are stored in large vacuoles, and then during the day, the stomata are closed for water conservation, and we'll have NADP and ATP available because that's going to drive the light-dependent reaction. So that C4 molecule then will release that CO2 to the Calvin cycle. We do see these. This is common of our bromeliads. Also in our desert plants, bromeliads are the pineapple family. Basically at night, carbon fixation does occur. And then during the day, when you have light energy to drive the light-dependent reactions to make ATP and NADPH, that's when we'll have the CO2 come into the Calvin cycle there to make our G3P molecule. So the different methods of photosynthesis each have advantages and disadvantages. It really depends on the climate. C4 plants are most adapted to high light intensities, temperatures with limited rainfall. C3 plants are better adapted to cold, below 25 degrees Celsius with higher moisture. And then our CAM plants are better adapted to extreme aridity. Basically, CAM occurs in 23 families of flowering plants. Remember, flowering plants are angiosperms, and they are also found among some of our non-flowering plants as well. So that's it for photosynthesis. That was our whole chapter covered in about a 40-minute lecture. Have a great day, everybody.